Hi, I'm Sam Hoare from Pharmacology. In this video, we're going to talk about measuring the binding of molecules to targets, specifically the data analysis artifacts that can occur in these experiments. Molecular interactions are quantified by the potency, which is the concentration of molecule required to produce a specified effect, usually 50%. The pharmacology data analysis we use to measure potency is around 100 years old. The analysis makes certain assumptions appropriate for the assay systems of the time. First, it was assumed the target concentration was really low. Second, it was assumed that the binding interaction was at equilibrium. However, in the modern era, these assumptions are often broken by the assays we are using in the lab. As you'll see, this happens with some of our most exciting new assay technologies and also with our most attractive molecules in drug discovery. Today we're going to talk about the fundamental assumptions of pharmacology data analysis, see examples of when these assumptions are broken, and go through solutions to these issues. This video emerged from a workshop I have done with the NIH Assay Guidance Manual. The AGM is a translational science, education and training initiative from NCATS at the US National Institutes of Health. This guide and associated workshops provide scientists with invaluable information for the design of bioassay experiments. For more information, check out this article in Nature Reviews Drug Discovery. You can access these links from the slide deck, which is available from the link in the comments. The system we are dealing with here is simply ligand interaction with a target. Here in blue we have the target. This can be any type of target: an enzyme, receptor, kinase, ion channel, transcription factor, and so on. Then we have the ligand here in red. This is the collective name for anything that binds the target to produce the desired biological effect: an enzyme inhibitor, a receptor agonist or antagonist, an antibody, ion channel blocker, and so on. The ligand interacts with the target to form the target-ligand complex, as we can see here. In our assays, we are measuring the level of this target-ligand complex, either directly or indirectly. We vary the concentration of ligand on the x-axis here and measure the effect on the target on the y-axis. A couple of things to note about the x-axis. This is typically the total concentration of ligand added to the assay, and this detail is very important. Also, the x-axis is typically logarithmic, which makes this curve sigmoidal. Now we analyze the data to determine the potency of the ligand on the target. We fit the data to a sigmoid curve equation, and from this curve fit, we determine the potency. Potency is the midpoint x value on the curve here. More precisely, the concentration of ligand that elicits 50% of the maximal effect. The potency is 1 nanomolar in this case, as you can see. This graph is for the simplest type of assay, a direct ligand binding assay. In such an assay, the potency is the affinity, or the KD. Please note, however, that the concepts we're talking about here apply to all bioassays that quantify ligand potency, be it EC50, IC50, Ki, and so on. In this video, we're going to talk about how modern assay conditions affect the reliability of this data analysis. Now, let's talk about assumptions underlying this analysis. The first assumption is that the target concentration is much less than the ligand concentration. In other words, the ligand is in large excess over the target. The second assumption is that the target-ligand interaction has reached equilibrium. This means the binding kinetics are fast relative to the incubation time. Now, in the classical era of pharmacology, These assumptions were almost always true, to such an extent that we have kind of forgotten about them. In the modern era, things are quite different. The assumptions are very often infringed, resulting in incorrect potency estimates. Here we are first going to talk about the target concentration effect, since it is quite simple. In the second half of this video, we'll talk about equilibrium, which is a bit more complicated. In the olden days, the concentration of target in the assay was always really low, in the femtomolar range, as represented by this diagram here. On the right is a target occupancy curve for a target concentration of 30 femtomolar and a ligand with a binding affinity Kd of 1 nanomolar. Target density was low because we were using tissues, so the expression was at the low level we typically find naturally. In addition, the volume of the assay was really high, in the milliliter range. Assays were run in test tubes or organ baths. These conditions meant that the target concentration was really low, and much lower than the ligand concentration in the assay. We quantify potency as the ligand concentration at the midpoint X value. We call this the L50 value. If our assumptions are being met, then the L50 is equal to the Kd, and that's what we see here. Both values are 1 nanomolar. In the modern era, the target concentration can be much higher, as illustrated in the diagram here. We express targets at artificially high levels and often purify them. Assay volumes are now in the microliter range. These conditions result in increases in the concentration of the target. In this graph, the blue line represents the same interaction we saw in the previous slide, but at a higher target concentration of 20 nanomolar. This significantly affects the data, as you can see. The increase in target density shifts the curve to the right by an order of magnitude. Now the L50 we determine is 11 nanomolar, which is 11-fold greater than the actual Kd value of 1 nanomolar. The L50 no longer matches the Kd. This means that a ligand with 1 nanomolar affinity looks like a ligand with 11 nanomolar affinity. This is bad news because we are reporting the wrong value of the ligand affinity for the target here. In some experiments, we deliberately use a large amount of target, for example, in DNA-encoded library screening. In this example, the target concentration is 200 nanomolar. Under these conditions, the binding curve is dramatically shifted. In this example, the L50 value is 100 nanomolar. In other words, a ligand with 1 nanomolar affinity looks like a ligand with 100 nanomolar affinity, and that's kind of scary. While these ligands can be really good for finding hits, they are problematic for quantifying the potency of the ligand. The target density problem has happened in part by accident due to advances in technology that have dramatically increased the target concentration in our assays. First, we have increased the target density in the biological material we use. Instead of the target being sparsely distributed in a tissue, now the target is densely packed in engineered cell systems and purified preparations. In this experiment here, we measured the expression level of a receptor, the CRF1 receptor. In native tissue, the cerebellum, the level was 0.23 picomoles per mg. In engineered HEK cells, the level was 62 picomoles per mg. This is 270-fold higher. Note this y-axis here is logarithmic. Second, we have decreased the assay volume. In the olden days, we used milliliters of assay volume in organ baths and test tubes. Now we use microliters of volume in microtiter plates. This is a change of 1,000-fold. This matters because when we decrease the volume, we increase the target concentration. These changes combine to produce very large increases of target density, by 5 orders of magnitude or more in some cases. The target density issue has also happened by design. Certain screening experiments actually work by using a huge amount of target in order to soak up the ligand. There are new assay technologies that employ target immobilized on a column or on beads. These include DNA-encoded library screens and also affinity mass spec assays, where mass spec is used to quantify the ligand. Here we load the ligand, and the goal is to trap the ligand on the column. This is done by having a high concentration of target packed onto the column. Target concentrations in the hundreds of nanomolar are usually encountered here. While these assays are great for finding hits, they are usually not suitable for quantifying ligand potency owing to this high target density, and we will see an example of this soon. Let's take a closer look at what happens to the ligand binding curve when we have a high target concentration. To do this, we're going to use simulated data. We created a simulator in Excel that you can see here. We made this simulator freely available to everyone, and you can find it in the link in the comments. You can use the simulator to explore the target concentration effect in your assays. It uses an equation that incorporates the target concentration into the analysis. You just plug in the relevant parameter values and it generates the curve, as you can see. The simulator also generates a data table shown here containing the values used to generate the graph. Here is the simulated data where the ligand affinity for the target is 1 nanomolar. We start with the ideal conditions, a target concentration of 30 femtomolar. Here the L50 is 1 nanomolar, so the data are what we expect. Now let's increase the target concentration to 20 nanomolar. Two things happen. First, the curve is shifted over to the right as we saw earlier. Second, the curve shape is distorted. It gets a lot steeper, and if you look closely, it's no longer symmetrical. These distorted curve shapes can be indicative of a target density artifact. Now we also see this in practice, and there's a really nice example of this in a paper from Stephen Charlton's lab. As we increase the target concentration from 42 picomolar to 1.5 nanomolar, the curve shifts to the right and gets steeper. Here's another example employing new assay technology. Recently, a colleague was working up an affinity mass spec assay for a target. In this assay, the target is immobilized on a column and binding of the ligand determined by mass spec. We were wanting to validate the assay by testing the affinity of a range of known ligands for this target. We went to the literature and found a set of ligands with a really nice spread of affinity as measured in a conventional radioligand binding assay. Here the range of affinity is around 1,000-fold. The graph here shows what we expect to see in a standard assay based on these affinity values, showing the large spread of affinity. Then we realized the concentration of the target on the column was really high, in this case 400 nanomolar. Before doing the experiment, we reran the simulation to see what the data would look like with this high target density. This is what we saw. Clearly, this is not reporting the affinity correctly. The L50 of these ligands is effectively the same. This is because of the high target concentration in the assay. Here we learned something. Having the information on the target concentration was really useful because it stopped us from wasting our time doing an experiment that wasn't going to work the way we wanted. This example highlights the difficulties we can encounter when applying old pharmacology to new assay technology without thinking about the assumptions of the pharmacology analysis. One final point here is the limit of sensitivity. The lowest L50 we can detect in this assay is 200 nanomolar, indicated by this red line. This limit is half of the target concentration of 400 nanomolar. This is a general rule. The lowest L50 we can measure is half the target concentration. Now, at this point, we could reasonably wonder, what the hell is going on here? It's not supposed to work like this, right? Well, the reason is another bit of pharmacology theory that we have largely forgotten about. We need to consider something called the free concentration of ligand. Up until now, our ligand concentration has been the total concentration added to the assay. We add 1 micromolar ligand to the assay and use that total concentration value in our analysis. Let's look at this total concentration. In this diagram, we have a population of ligands in red and targets in blue. The total concentration is all the red circles you can see. We can split this total ligand concentration into two subpopulations. Some of the ligand molecules are free in solution. This is the free concentration of ligand. And then some of the ligand molecules are bound to the target, which is the bound concentration. Now, let's view the total concentration of ligand added to the assay by fading out the targets. Now, let's look at the free concentration. Here's the same set of ligands and targets as before. Now we're going to visualize the free concentration of ligand. To do this, we fade out all the target ligand complexes. This shows that the free concentration of ligand is a lot less than the total concentration when we have a high target density. This phenomenon is called ligand depletion, and you will see this written about in pharmacology textbooks. Ligand depletion is the reduction of the free ligand concentration that results when the target binds up a significant fraction of the ligand that we add to the assay. Now, here's the pharmacology fine print. It turns out that the affinity, or KD, of a ligand is the free concentration of ligand required to occupy 50% of the targets. The KD is not the total concentration of ligand we add to the assay, it's the free concentration of ligand that remains in solution after ligand has bound to the target. In the previous slides, we have effectively been plotting the wrong value on the x-axis of our graphs. Instead of using the total concentration, in principle, we should be using the free concentration. On this slide, we're going to see what happens when we use the free concentration on the x-axis. On top here, we have our data we saw earlier with the total ligand concentration on the x-axis. The ligand affinity is 1 nanomolar and the target concentration is quite high at 20 nanomolar. The curve here is right shifted with an L50 of 11 nanomolar owing to the high target concentration. Now let's calculate the free concentration. We're going to use this data point here where the total ligand concentration is 10 nanomolar. The target ligand complex concentration here is 9.2 nanomolar. This value is from the simulation spreadsheet we used to generate the data. Now we can calculate the free ligand concentration. We simply subtract the target ligand complex from the total ligand. That is 10 minus 9.2, which gives us a free ligand concentration of 0.8 nanomolar. Note this free ligand value of 0.8 nanomolar is much less than the total ligand value of 10 nanomolar. Now let's replot these data in terms of the free concentration of ligand, which is the x-axis on this bottom graph. Here is our data point for 10 nanomolar shifted over to 0.8 nanomolar, when we use the free concentration. We calculated the free concentration for all the data points on the graph, then fit the curve. You can see the resulting curve on the bottom is shifted to the left relative to the curve on the top. The measured Kd from the curve fit is 1 nanomolar, which is the actual Kd of the ligand. Things are working as we expect in the bottom graph because we are using the right value on the x-axis, the free concentration of ligand. In some cases, we can measure or calculate this free concentration of ligand. For example, in mass spec assays, we could, in principle, measure the free concentration flowing through the column. Here we apply the ligand and most gets trapped because of the high target density. However, the free ligand comes through the column. If we can measure this free ligand concentration, then we can use this value on the x-axis of our binding curve. We recommend this approach if it is technically feasible. So, what are we going to do about this problem? The first thing is simply awareness. Be mindful that your assay has a limit of sensitivity for quantifying potency based on the concentration of target. The lowest L50 we can detect is 50% of the target concentration if we're using total ligand concentration on the x-axis of the graph. In order to know this limit, try to calculate or at least estimate the target concentration in the assay. This will help you to know if the target density is going to be a problem. The simplest way to reduce the target concentration is to increase the volume. For example, if we're using high-density plates for HTS assays, we can increase the volume by using a 96-well plate. This can enable a 50-fold increase of the volume and so a 50-fold decrease of the target concentration. Sometimes, if we need to, we can go to extremes. We can do assays in deep well blocks to increase the volume to 1 ml. I've even done a radioligand binding assay using a 10 ml assay volume to avoid the target density artifact. The second thing we can do is to reduce the amount of target. This is doable, but it can be a lot of work because it can involve changing the technology. For example, oftentimes we use GFP as a fluorescent label for our proteins. This has some very good properties, as you know, but the emission intensity is quite low for GFP, meaning we often need a lot of protein to see a signal. We can reduce the amount of protein by using a brighter fluorophore. In this case, we tagged the target with an FITC labeled antibody. FITC has a much higher emission intensity than GFP. This means we can reduce the amount of target, sometimes by 100-fold or more. One potential solution is to use the equation we saw earlier that explicitly takes into account the target density. In theory, this is a really elegant solution. However, in practice, it just doesn't work very well because of experimental variability. A small amount of variability transmits to a large error in the estimate of potency using this equation. For example, here the fitted error of the Kd using the equation is pretty large relative to the measured Kd value. In addition, we need a really precise measurement of the concentration of target in the assay. Small errors in the target concentration can result in large errors in the ligand affinity estimate. Often we don't know with precision the concentration of target in the assay, and this creates large errors in the affinity estimate when using this equation. I'm sorry, the news isn't better here. Here is a summary of recommendations for the target density issue. First, just be aware that target concentration can affect your potency measurements if there is a high target density in your assay. Second, attempt to estimate the target concentration. Even a rough estimate will give you an impression of whether the target concentration could be affecting your potency data. Then we come on to the solutions if we need to reduce the target concentration to get better potency measurements. The easiest thing is often just to increase the volume, especially if we are coming from an HDS screen. Next, if feasible, we can decrease the target amount by increasing the assay sensitivity. Now we will move on to the second assumption that can impact our data analysis. This is the issue of equilibration and the assay incubation time. Classical pharmacology analysis assumes the assay has closely approached equilibrium. This means that the assay has been incubated long enough for the level of target ligand complex to closely approach the plateau of the time course. We can explain this using the graph here, which shows the familiar association curve describing the increase of target binding over time. At the plateau here, the binding interaction has effectively reached equilibrium. Our pharmacology data analysis assumes we have reached this point in our assays. In some modern assays, we are not at equilibrium and instead are further back on the association curve down here on the rise phase. This means we are underestimating occupancy of the target by the ligand. This in turn means we will underestimate the potency of the ligand for the target. The bottom line here is if we don't incubate for long enough, our potency measurements will be wrong. To understand this issue, we need to consider the kinetics of ligand binding to the target. Here is the familiar diagram of binding kinetics. The ligand associates with the target, defined by the association rate constant, or K-on. After staying bound for a time, the ligand then dissociates from the target, defined by the dissociation rate constant, or K-off. This dissociation is usually expressed as the half-time, which is like radioactivity. It's the time it takes half the target ligand complexes to break down. We want to know how long it takes equilibrium to be reached. This can be calculated from the kinetics. It turns out the equilibration time is dictated by the dissociation rate constant K-off. That's not a typo, it is the dissociation rate. This is explained in these articles here, and I'm just going to ask you to trust me on this. It turns out that equilibrium is closely approached within 3 times the dissociation half-time. This is a really useful equation because it tells us how long we need to incubate our assays for if we know the K-off value. Let's examine this effect on the measurement of ligand potency. Here we have our assay where we measure target occupancy by the ligand on the y-axis at varying concentrations of ligand on the x-axis. We assume we don't have a target concentration issue in this assay, so we don't have that complication here. Let's assume the ligand affinity for the target is 1 nanomolar. First, let's assume the assay is at equilibrium, which is shown by this black line on the graph. You can see the L50 is at 1 nanomolar, matching the KD as expected. Now, this compound takes an unusually long time to equilibrate. The equilibration time here is 12 hours because the dissociation half-time of the compound is 4 hours. This slow equilibration can happen with high affinity ligands, as we shall see, owing to this slow dissociation. Here's what happens if we use a short incubation time of 10 minutes. This short incubation is a problem here. The binding curve is shifted to the right, so we are underestimating affinity. Occupancy of low concentrations is underestimated because the assay has not reached the equilibrium plateau and is instead further down on the association curve, like we saw on the previous slide. The effect is quite dramatic. The L50 here is 20 nanomolar, which represents a 20-fold underestimation of the ligand affinity for the target. Here's what happens if we use an incubation time of 1 hour, a time that is fairly typical. Again, the curve is shifted to the right. The effect is not as dramatic, but we are still underestimating ligand affinity by 4-fold here. Here's what happens with a longer incubation time of 6 hours. This long duration is often feasible with some of the more robust assays we use in drug discovery. Here you can see the performance is better. The curve pretty much overlies the equilibrium curve. The extension of the incubation time is a simple trick to detect whether there is an equilibration issue with our compounds. Now let's look at this issue of short incubation times with a real-world example. In the early 2000s, a new technology emerged for assaying G-protein coupled receptors. We could use biotechnology to force these receptors to couple to calcium signaling in the cells. We could measure calcium signaling quite easily using the flipper plate reader. This was good for a number of reasons. We could use a common assay platform using the same instrument for all our receptors, which dramatically improved efficiency, as you can imagine. Calcium assays are inexpensive, so we could test thousands of compounds, which was great. This worked really well for high throughput screening, where we weren't too concerned with quantifying ligand potency. However, in our lab and many others, we ran into a major problem when using the calcium assay for SAR in lead optimization. Here we wanted to measure the potency more accurately. Chemists started to notice an awful lot of flat SAR, meaning we made chemical changes to the molecule that did not produce any changes to ligand potency. Here's an example of flat SAR from a paper from Craig Lindsley's lab at Vanderbilt, who were using the calcium assay. This shows the SAR reaching a potency in the 200 nanomolar range, but then getting stuck there. No matter what the chemical modifications, the potency was not increased here. We saw this in our lab many times with the calcium assay. In lead optimization, the compound potency kept hitting a ceiling around 100 nanomolar that we could not seem to break through. This was really frustrating, as you can imagine. We suspected the problem was with the assay rather than with the medicinal chemistry. It turns out the problem was the incubation time. Calcium responses are really fast. On the left is a time course graph for a fairly typical calcium response stimulated by a GPCR. The peak response here is within 10 seconds of drug addition, and we use this peak time as the time point for our SAR assays. This meant that the assay was nowhere near equilibrium for most of the compounds we were testing. This effect was evaluated by numerous investigators, and you can read the papers here if you want a deeper dive. The overall conclusion: avoid short incubations. We can explore this effect using simulations. Here we have an assay for 8 different compounds with a range of affinity, with the KD ranging from 0.3 nanomolar to 1 micromolar. In this experiment, the assay has reached equilibrium, and clearly we can distinguish between the compounds here on the graph. Next, we ran a simulation assuming an incubation time of 30 seconds, something we might use in the calcium assay we just talked about. This is pretty bad, obviously. The potent compounds are all bunching up in the 100 nanomolar range, and using this short incubation here, we have artificially flat SAR. We can't distinguish between the compounds, even though in reality their affinity for the target is very different. The other area where equilibration time can have an impact is for potent ligands. In general, the more potent a ligand is, the longer it takes to equilibrate with the target. This is because more potent ligands dissociate more slowly from the target. And remember, our equilibration time is dependent on the dissociation rate. Here we have some simulated target occupancy data for a series of compounds with a range of affinity. In this graph, we assume the assay is at equilibrium. We have some potent compounds here, with the KD going all the way down to 0.01 nanomolar, which is 10 picomolar. This high affinity is a result of a slow dissociation rate. For example, the highest affinity compound here has a dissociation half-time of 12 hours. This means the highest affinity compounds take a long time to equilibrate, as you can see here. Now let's rerun this simulation, but instead let's assume an incubation time of 1 hour. Here is what the data look like. You can see here the highest affinity compounds can no longer be discriminated from each other. The L50 values here are all bunching up around the 0.1 nanomolar range. This means we are not measuring affinity correctly for our highest affinity ligands. This can be bad news for drug discovery, because these high affinity compounds are often the ones we are selecting to take into clinical trials. Having the wrong affinity can lead to incorrect predictions of drug effect in humans and also in animal models, and we will see an example of this in a minute. One way to see whether equilibration time is an issue is to simply extend the incubation time. Let's extend the incubation time here to 6 hours. Here we can see this changes the binding curves. We can now discriminate between the highest affinity ligands. This change in the data on extending the incubation time can be a really useful diagnostic and can provide better estimates of affinity. The best solution to the equilibration issue is to abandon the attempt to reach equilibrium and instead measure the kinetics of binding. The kinetics are the association and dissociation rates, K on and K off. The trick here is we can calculate affinity from these rates. KD is simply K off divided by K on. These papers here describe methods for measuring kinetics. This video we made gets into the details of kinetics, including these methods. It's not straightforward, but the payoff can be considerable in terms of avoiding mistakes in selecting compounds and predicting human dosing. Here's a real-world example of this equilibration phenomenon. This was a drug discovery project I was involved in at Neurocrine Biosciences in partnership with GSK. We were developing antagonists for the CRF receptor, a GPCR. We had identified a compound that advanced to the clinic that unfortunately we had to discontinue for tox reasons. This compound is NBI 30775. Here is the in vivo data in rats for this compound. On the y-axis we have inhibition of release of the hormone ACTH, and on the x-axis we have the plasma concentration of the antagonist compound. We can see here nice strong efficacy of the compound in vivo. We were developing new molecules. To establish the SAR, we were using a standard in vitro binding assay with a 90-minute incubation at room temperature. In this assay, 775 bound with an apparent affinity of 10 nanomolar. We identified numerous compounds that were active in the binding assay, and here are listed their apparent affinity values. You can see they bind with a similar apparent affinity as 775 in the in vitro assay. We then tested the compounds in vivo, and this is what we got. Clearly, these new molecules were much less active in vivo, and we were quite shocked to see this. Some of these compounds had much higher plasma concentrations than 775, but were still considerably less active in vivo. At this time, we were evaluating the kinetics of the compounds, and we discovered something. Under the conditions of the in vitro assay, 30775 dissociated really slowly, with a dissociation half-time of 2 days at room temperature. The equilibration time then would be 6 days. Consequently, using our standard 90-minute incubation, this compound was very far from equilibrium, and so this affinity value here is almost certainly wrong. We developed kinetic assays to quantify the Kd from the rates, as mentioned on the previous slide. We sped things up by increasing the temperature to 37 degrees. Under these conditions, the true affinity of 775 was found to be much higher than under our original conditions, with a Kd of 0.8 nanomolar. Now we use the kinetic assay to quantify the new molecules. The true affinity of the other compounds turned out to be much less than the affinity of 775 by 100-fold in some cases. This provides a simple explanation for why the compounds were less potent in vivo. They were simply less potent on the receptor when we did the assay correctly. Next, we use this new assay to identify new molecules. Here is an example with an affinity of 0.3 nanomolar. We were pleased to see this compound was really active in vivo. Compounds like this have now advanced to phase 3 clinical trials for treating congenital adrenal hyperplasia. Here are recommendations for the equilibration issue. First, avoid short incubations if you can, or at least be aware of the limits if you do use these conditions. Second, for high affinity ligands, be aware that conventional assays may not be at equilibrium for these compounds. We can test for this by extending the incubation time to see if the potency goes up. Ultimately, it's a good idea to understand the kinetics of compounds advancing to the clinic. This is especially advised if there are surprises in the efficacy of compounds in animal models. In summary, we have entered a new world of assay technology and ligand pharmacology. We need to be mindful that some of our coolest new technology and most exciting drug candidates could well violate assumptions of the analysis we use, leading to incorrect measurements of ligand potency. There has been much discussion of this in the literature, and here are some papers to get you started if you'd like a deeper dive. I'd like to thank some people who made this video possible. Serene Markosian, editor of the Assay Guidance Manual, works tirelessly to put workshops together for which this material was originally prepared and presented. And thanks to Jamie Darlin, who is a pioneer in assay quality control and a leading expert in artifacts, who encouraged the work you've seen today. Thanks to Mark Navarre, who has been helpful in providing some real-world insight into the issues we have seen. And finally, thank you all for your interest and attention.